Okay, good. So let's shift gears here for the remainder of the course. We are going to be taking a look at two, I think, very interesting areas of cancer biology that we don't typically think of when we're really focused on the more sort of obvious, attractive things like oncogenes and tumor suppressor genes, right? And that is the role of the immune system in preventing cancer, and a reminder that we actually depend upon our immune system very heavily in terms of making sure that cancer is not developing in our bodies throughout our lifetime. Right. We will also look at clinical examples of how we can treat cancers using different types of therapeutic strategies, including different types of exciting immunotherapies. Right. So the remainder of our class will be focused on these two sort of broader topics that I think you'll find very interesting. So in order for us to have a meaningful discussion about the role of our immune system, we need to make sure that we're all on the same page and at least have a fundamental understanding of how our immune system works. I think many of you have taken immunology already, right? But I think some of you have not. And so what I'll do today is just give you guys a refresher course for those of you who have already taken immunology. And for those of you who haven't, it's kind of like a crash course, right, in the important concepts involved in our discussion so that you can actually understand when we talk about what's going on with cancer and our immune system. So our immune system, the two sort of key features of our immune system of relevance here are specificity and memory. Right? What do I mean by that? The ability of immune cells and components of our immune system in our body to be able to specifically identify threats. Right? And memory, the ability of our immune system, once it has learned about a novel threat, to be able to respond in the future, repeatedly and effectively, with the ability to repeatedly recognize this as an ongoing problem. A key concept in the ability of our immune system to work properly is the ability of our cells in our body, the immune cells, to be able to distinguish between certain signals that are self signals, meaning normal, everyday, different types of molecular shapes that are present as part of your body's normal functioning, right? All the cells, tissues, whatever, right, that have thousands upon thousands of different normal human proteins that might be a part of them, right, in your daily life. To be able to distinguish those types of signals as being self from signals that are non-self, right, signals that are wrong or bad, foreign, right, that might represent a way of identifying something as a threat. As a whole, our immune system has three major functions. One is to constantly be maintaining the health of our tissues through recognizing and removing abnormal self cells. Secondarily, our tissue, as part of its sort of janitorial or custodial activities, have to be able to remove dead or damaged cells, right? Cells that are constantly dying off as part of their normal cell cycle, right? They have to be removed because otherwise, they would just build up in your body. And third is our immune system is focused on protecting against invaders like pathogens. Now, I think typically when you ask somebody, what's the job of the immune system, right, they would probably just say, oh, it's to protect against invaders, right, like bacteria or viruses or parasites. I think very little regard is given to the other two tasks, but I've intentionally listed those tasks first, because if you think about this, on a typical day, as you're sitting there right now, participating in our class lecture, the immune cells in your body are bumping into way, way more normal self cells than they are bumping into, let's say, virus particles or bacteria. So therefore, the non-pathogenic component of our immune system, right? In other words, the component that's mainly concerned with tissue maintenance and health, is actually far more representative of the activity of our immune systems. The occasional invasion by a pathogen, whether you get a little cut or you eat some food that's contaminated or you breathe something in that has maybe virus or bacteria, right? That represents a relatively rare occurrence or a relatively rare encounter, right, for a given immune cell compared to the multitude of normal cells in your body that it's encountering all the time. Right? So the first one here, recognizing and removing abnormal self cells, hopefully you can already realize, yes, that is the link that we're talking about here between the immune system and cancer cells. So in order for transformed cells that are becoming cancerous to make it all the way to the point where, like in the last few lectures, we've been talking about solid tumors that have reached a critical mass where they undergo metastases, right? One thing that you have to think about is throughout this long drawn out process that led maybe for many years to there being a development of a tumor, and then pretty rapidly in the span of a couple years, that tumor becoming aggressive metastases, all through that history, right, in the background, somehow these cancer cells were avoiding or eluding the immune system. Right? So let's think about different ways that this can happen. So first, let's do our little crash course review, right, of cells that are involved in the immune system. So primarily when we talk about fighting against transformed or cancerous cells in our body, we are talking about the involvement of white blood cells in our immune system, the leukocytes. So there are a handful of different types of leukocytes represented here. Several of them are the types that you would find floating around in your blood at any given time. Certain types are only found sort of resident in tissues. For example, monocytes. Monocytes are these large white blood cells that have a very big nucleus, right? These are the ones that float around in your blood, but when they arrive at certain sites in your body's tissues where they're needed, they can undergo diapedesis. That means they squeeze out of the blood vessels, and they can fully differentiate into macrophages, right? And then the macrophages are now crawling around doing cleanup, right? Also, we have things like lymphocytes. Certain types of lymphocytes, like B cells, can differentiate further into plasma cells to produce antibodies. We also have cells like these granulocytes on the left that produce and store many types of sort of noxious types of compounds or molecules, signals that, if they are released, can cause things like inflammation or potentially be used to fight against certain types of pathogens, right? Because these different types of granulocytes store these different molecules in these vesicles that stain differently depending upon if you use basic stain or acidic stains, right? Or neutral stains. That's historically what gives them these names, like basophils that stain with basic stains, eosinophils that stain with acidic eosin, right? And then neutrophils that stain with neutral, pH neutral types of stains. So these fall under the category of granulocytes. These over here fall into the category of phagocytes, meaning they work, macrophages work, for example, by ingesting or taking in, through phagocytosis, different types of targets like bacteria or debris from dead cells that need to be recycled. Right? Neutrophils kind of function as both. These cells can also be divided in terms of how they carry out their function. So for example, certain types of cells are cytotoxic. So certain lymphocytes like T cells, killer T cells, work to kill other target cells by releasing certain types of small molecular signals, or presenting different types of death signals to receptors on the target cell. Right? So they function in a cytotoxic way. A subset of these cells also have the ability to function as professional antigen-presenting cells. What is an antigen? An antigen simply is a molecule that has a particular three-dimensional shape that might be suitable as a unique three-dimensional shape that the cells of your immune system can learn to recognize, right, as being something worth paying attention to. So these cells over here can be considered professional antigen-presenting cells because not only do they carry out other tasks of being phagocytic or cytotoxic, right, but they also present evidence of different antigens that they are collecting to be able to pass the message on to other immune cells that something is going on here, and these are the types of antigenic targets that are involved. Dendritic cells, sort of the lowliest of the leukocytes, right? They're not terribly exciting because whereas things like granulocytes can sort of explode and release all kinds of histamines and other kinds of exciting molecules like that, and then you have phagocytes that eat things, right? Lymphocytes that can kill other cells. Dendritic cells, they just roam around like little scavengers and they just pick up bits and pieces of things. They phagocytose little scraps of things, and they just present on their surface a whole bunch of different antigens that they've found so that they can facilitate informing other immune cells that bump into them about what types of things the dendritic cell is finding. So these cells vary in population. So you can see the most popular cells in your blood. So this is just looking at the percentage of these types of white blood cells that are circulating in your blood. Right? Most abundant are the neutrophils, right? Followed by things like the lymphocytes. Then you have the monocytes, right? That might turn into macrophages once they get into the tissue. And then finally, the more rare ones like the eosinophils, and the rarest are things like basophils and mast cells. So the lymphocytes, there are two main types, right? Or I should say there are three main types, sorry. There are the B cells, the T cells, and the natural killer cells, okay? And you don't have to memorize this table. Hopefully, for those of you who took immunology already, all of this rings a bell, right? And hopefully you're like, hey, yeah, that's right, I know this, right? For those of you who don't or have not taken immunology, right, I just want you to be familiar with the fact that these are the functions of these different types of cells down here, right? Okay, so let's discuss this. How does our adaptive immune system work? So remember, it's our adaptive immune system, the ability to recognize something new, recognize it as a target, not only eliminate it, but also have memory of it, right? So that you don't have to go through this whole relearning process every single time. So the first time you're exposed to something that's novel, that's different, for example, I should say, in our everyday lives, we are, of course, exposed to a multitude of different types of microbes, right? There's bacteria on the surface of your desk. There's bacteria on the surfaces of the handle of a doorknob, right? There's bacteria all over your own skin. Most of that stuff stays outside of our body because we have good physical barriers to keep them out, like our skin and our mucosal layers of our GI tract or our respiratory tract, for example, right? Even if certain bacteria do get in, for example, you get a paper cut, right? Even if it's not a bad paper cut, you're like, oh, right? Whatever bacteria that was on your skin around that cut, now it gets in. The majority of everyday types of pathogens, like common everyday bacteria, right? Our immune system is already kind of preset to recognize because certain types of antigenic signals on common everyday bacteria and viruses, because they've evolved with us over eons, hundreds of millions of years, right? It makes sense that as part of the human immune system developing, right, we've already come preset with the ability to recognize certain types of generic pathogenic features. right? So we don't actually need to have a big, elaborate, adaptive immune response to learn something new, right? But let's say, in this case, you have a bacteria like this red one here that is something new, meaning that it's something that your immune system does not come pre-programmed to recognize. So in this case, right, this bacteria might start multiplying in your body and makes you sick, right? But on its surface are novel antigens. And so you may have something like a B cell, a B lymphocyte here, that has on its surface specific immunoglobulin proteins, or Igs. And these are designed to have unique three-dimensional binding arms that may, by chance, bind to this particular antigen, right, on the surface of this particular pathogen, right, this bacteria. If that's the case, this particular B cell, B lymphocyte, will suddenly become pretty valuable because it actually looks like it works to detect something. And so it will undergo rapid clonal expansion. So you make many, many copies of this valuable B cell. Some of these B cells will be told to differentiate into plasma cells. And these plasma cells will now continue to make that immunoglobulin protein that's on its surface that it uniquely has, right, as part of being this particular clone of B cells. But importantly, the plasma cells will make the soluble or free-floating version of these immunoglobulins that we call antibodies. Right? So you can see here, this is a little chart showing that over the course of weeks following your first antigen exposure to this nasty bacteria or whatever that made you sick, so your built-in preset innate immune system doesn't succeed in eliminating it, so that's why you get sick, right? Usually it takes about a week or so before you start developing really strong symptoms, and your adaptive immune response, all these B cells, then have to be mobilized, right? And they have to start going to work in order to now save your life, right? So really, it takes a couple weeks following first antigen exposure before you have a high level of antibodies in your blood, right? So up here, these are the green antibodies, right? Because it takes that long for this process on the left to happen, selecting for B cells and having them turn into plasma cells. And importantly, even after you beat the infection, right, these antibodies stick around for a while to continuously give you protection. And that's part of your memory, right? Another important part of your memory is, remember, these antibodies are made by plasma cells, and these plasma cells come from B cells that are told to turn into plasma cells. So in order to have long-term memory, you need to have some of these B cells stick around, right? These B cells that have the right immunoglobulin on their surface. So some of these become memory B cells that stick around, like this one on the bottom of the slide. So now we'll continue down this slide and start from the top on the next slide. So all of this on this slide is your primary immune response. Right? Your secondary immune response, so here's that same memory B cell that stuck around, right? The secondary immune response happens the second time or any subsequent time you're exposed to that same pathogen again. It could be days later, it could be weeks, months, or even years later, right? You encounter that same pathogen again. Fortunately, because you have these memory cells, you don't have to start from square one again, looking for or selecting for suitable B cells, right? You already have these really good B cells that have stuck around that were selected for previously because they have good immunoglobulins on them. So that means during a secondary immune response, right, these B cells very rapidly multiply. And you can see here, you have a huge production of antibodies very quickly, right? And if this works, then you don't even get sick from the bacteria or the virus again, right? Because your immune system works so fast now because it recognizes it that you don't even give time for the virus or the bacteria to multiply and cause problems, right? Okay, so when this stuff is happening, what are the different sort of participants in terms of different cells that are carrying this out? Let's say on the left, you have a virus that invades a host cell. I know these last couple slides we're talking about viruses and bacteria. You're like, I thought this is a cancer class, right? Yeah, but for the purpose of this sort of crash course review, it's more useful and simplistic to think about viruses and bacteria first, right, as being the things that are presenting these antigen targets of interest. So these bacteria, they infect a host cell in your body, right? So the cell is getting hijacked, right? So this poor cell was just minding its own business as a respiratory epithelial cell, and now it's been hijacked, right? It's producing potentially more virus particles. So the cells in our body, as part of their normal physiology, present on their surface examples of different peptides that are coming from the proteins inside of them. This is because different immune cells that are circulating around, checking on different cells, can't look inside of a cell to see if there's anything wrong with it. Right? Something like a macrophage or a lymphocyte that's coming along and bumping into one of your body cells, right? It can't actually peer into that cell to see what's going on. Therefore, it relies upon that cell to actually show or exhibit what's going on inside of it. Right? So all of the nucleated cells in our body do this to varying degrees. Right? They do this because all cells in our body, to varying degrees, produce these MHC platform proteins. So in this case, it's this little pink rectangle over here. And this serves as a little platform on which it will present examples of different small peptide fragments that are coming from proteins inside of the cell. So in this case, the red triangle represents a little fragment of some protein that comes from the virus, right, that is now inside the cell causing trouble. So by presenting it on this MHC platform, you can look on the right here, MHC platforms, they stand for major histocompatibility complex. Right? They are protein structures that hold onto and display different types of peptides. So inside the cell, let's pretend there are 4 proteins in here. Let's say 3 of them, the brown, the blue, the green one, are totally normal proteins that are part of your totally normal, healthy cell function, right? Those bits and pieces from those proteins get displayed also. In a way, it shows, right, that the cell has normal examples of proteins from inside of it. But now maybe this red one is a protein that came from the virus, right? And so this one then gets presented over here. So if you have immune cells that recognize this red dot, this small peptide, as being something foreign and different, right, then maybe that's a way for your immune cells to recognize that something funny is going on inside of this cell. Right? So there are 2 different types of platforms, MHC1 and MHC2. So MHC1 platforms are the one on which they present things that are coming from, let's say, an infection taking place. Right? They present all kinds of normal protein fragments from inside, as well as any abnormal protein fragments that are kind of all mixed up inside of the cell as a result of infection. So we go back to the left here. This infected cell is presenting the little red triangle on MHC1. That is what this cytotoxic T cell is able to recognize using its T cell receptor. And this causes the cytotoxic T cell then to activate and tell the infected cell to undergo apoptosis. So this is an example of extrinsically triggered apoptosis, right? So that cell hopefully will self-destruct, right? Remember, it pushes the nuclear button. There's a big explosion. Well, not a big explosion. I like a big explosion. The cell actually basically implodes as part of this carefully orchestrated apoptosis. And the whole point is, hopefully this happened before, right, it released too many more of virus particles, right? So this is the way to try to stop the infection. Another thing that can happen, let's head off to the right over here in the figure. If you already have antibodies floating around in your system that can detect the antigens from the virus, they will bind to the virus and make it much easier for things like macrophages to be able to now bind to the virus and consume it, phagocytose it, right? The macrophage will destroy the virus inside, and now the macrophage has an opportunity to display the little peptide fragments that are coming from the various proteins from the digested virus, right? And so it also displays it on MHC. Oh, but not so fast, right? It's not the same MHC it displays it on MHC2. See, it's a purple little rectangle here instead of the little pink MHC1 rectangle. Why? Right? Because this macrophage, when it's presenting the peptide, is not saying, help, I'm infected, I don't know what to do, right? What it's saying is, hey, I'm doing my job here, and this is what I'm finding, right? This is what I'm encountering when I'm eating this virus. So it is a professional antigen presenting cell that's presenting antigen to inform or pass the message on to other cells. So it itself doesn't want to be a target of being told to undergo apoptosis, right? Because that's killing the messenger, right? So in this case, helper T cells with their purple T cell receptors recognize and bind to the peptide that's being presented by the macrophage, right? And these helper cells then activate and they help. They help by coordinating the activity or the activation of more cytotoxic T cells so that these cytotoxic T cells can go out and look for the truly infected cells in your body and eliminate them. And these helper T cells will talk to the B cells that have the right immunoglobulin on their surface, right? So this B cell over here, its immunoglobulin is binding the virus as well, right? So this B cell is kind of excited, but this B cell is like, look, my immunoglobulins are binding to this thing, but I don't know what to do right now, right? So this helper T cell tells this B cell, oh, you should turn into a plasma cell, right? Because your immunoglobulin is what we need right now. Turn into a plasma cell and make tons of antibody that you know how to make, right? And so that is the ton of antibodies that then can feed back here, right? And that provides you the ability then to have a much stronger immune response. All right. So that was a whole semester of micro 461 immunology in like 2 slides, okay? All right, well, anyhow. All right, so let's move on. Let's bring cancer into the picture here, okay? Not surprisingly then, tumors, there must be some interaction between tumors and our immune system involving these types of immune functions that we just looked at, right? I think the striking thing that you must wonder then is, wait, yeah, if tumors are presenting all kinds of weird peptides and things, right, then they must be detected by our immune system, right? Yeah, they are. So when you have transformed cells, right, that are dividing and mutating, remember how we said the majority of those transforming cells as they divide in their daughter cells, right, most of them undergo apoptosis because they still have intact things like p53 signaling, so they can tell that there's something wrong with them, right? So p53 can pause the cell cycle, try to get some DNA repair enzymes to go to work. If the damage persists, like you have massive translocations that happen or loss of an entire arm of a chromosome, your cell's not going to be able to replace that, right? So that means the p53 signaling keeps going, and that leads to full arrest of the cell and then the triggering of apoptosis, right? So most of the transforming cells do that. Cells that don't do that, right, and continue on, right, then it's the job of your immune system to move in and say, nah, not so fast, right? You got weird signals on the outside. These are not signals that we recognize as being normal self signals. Therefore, we're going to shut you down by triggering apoptosis extrinsically. Right? So in order for you to actually get transformed cells that make their way and become a large tumor, right, somehow they are avoiding that second line of defense, the immune system. Look at these early experiments. I think this really nicely highlights this. Let's say you give 2 different strains of lab mice, a blue strain and a pink strain of lab mice, a big dose of carcinogens, right? Yeah, I mean, it sounds kind of mean, but this is what we do, right? And so these blue mice and these pink mice have different genetic backgrounds. That means their immune system profile is different, right? Different alleles, different variations of sequences in the various genes that code for the proteins that are used by immune cells to function. In both of these cases, they independently develop tumors as a result of that carcinogen. But these tumor cells, we'll just call the blue ones, right? Because somehow these tumor cells managed to stumble upon mutations that avoided the immune system of the blue mice, right? So only those cells that transformed and had the right combination of mutations to avoid the blue immune system, right? Those turned into the tumor in here. Likewise, the pink tumor cells are ones that stumbled upon mutations along the way and survived by learning how to avoid the pink immune system, right? If you take these tumor cells now and you take some of these cells and you inject them into both types of mice, what do you find? You find that the blue tumor cells can start growing again in the blue mice, but they don't grow in the pink mice. And vice versa. The pink tumor cells will continue growing, right? Start new tumors in pink mice, but not the blue ones. Why? Because these tumor cells have become so fine-tuned in their selection of mutations, right, to only be able to avoid certain types of interactions with immune cells that are from the blue background or the pink background. Right? You know, this is why, quick story, when I was a PhD student, right, in a prostate cancer lab, and my fellow grad students and I, you know, we'd have a lot of time to kind of wonder about things that were sort of not related to our projects. For example, you know, we worked with testosterone because it's prostate cancer cells, so we'd give cells testosterone. So as a result, we had access to a lot of DHT. And so we often thought like, hey, you know, what would happen if we just took this testosterone? You know, those are the type of stuff you think of, right, when you're a graduate student. We also were very excited because in the Sigma chemical catalog that we ordered different compounds from, you could actually order cocaine, right, from Sigma. Right? Yeah, I mean, it shouldn't be shocking because obviously there are labs out there who do studies on different types of, you know, drugs and things. So you could also order cocaine from there, but we never tried, right? But anyhow, the reason I say that is because one of the things we thought about is we're working with these prostate tumor cells, human prostate tumor cells all the time. And we kept thinking like, man, what if we accidentally exposed ourselves, right, through a needle stick or something to these human prostate tumor cells? Would they end up actually giving us prostate cancer, right? And of course, we realized, no, the answer is no, because those prostate tumor cells developed in somebody else's body, right? So those tumor cells became an aggressive tumor because their bag of tricks in terms of mutations was particularly selected for to allow them to avoid somebody else's particular genetic background of immune system, right? So the chances are if I accidentally poked myself with a syringe that had some of these prostate cancer cells, like if I'm trying to inject them in a mouse, right, and the mouse kind of squirms around and I accidentally jab myself, right? I know some of you are like, haha, you deserved it, right, for picking on that mouse. No, but the point is those tumor cells would be eliminated by my immune system, right? Hopefully. Fortunately, it never happened. Okay. So let's think about this other experiment. Let's say you have some tumor cells that have been irradiated, right? They're zapped with radiation to the point where even the tumors, even the cancer cells messed up genome becomes so messed up by the radiation that the cancer cell just dies and it doesn't function anymore, right? It cannot divide anymore, but it's still intact, right? So it has all kinds of antigens on its surface that are unique to that particular tumor profile. Let's say you inject some of these killed tumor cells from another mouse into this mouse, right? So these cells then, this mouse's immune system now has a chance, right, to see all of these different antigens, right? And the mouse has an opportunity to try to kind of come up with some sort of an immune response. Importantly, because these cancer cells are irradiated or killed, right, there's no chance of a new tumor forming. Right? So you give this mouse some time in order for its immune system to kind of make of this whatever it wants to make of it, right? And then later you take that same mouse and this time you inject cells again, but these are not killed tumor cells. These are now live tumor cells that came from the same original tumor where these killed cells were at the top, right? What happens? Well, what you find is that the mouse, because it had time for its immune system to kind of really figure out what's going on here and learn, you know, different antigenic targets from the tumor cells, the mouse is now protected, right? It actually has immune memory against this particular tumor cell. Right? But what happens if you take that same mouse that built up immunity against the red tumor, right, and you go ahead and now inject it with blue tumor cells that came from a different tumor, right? What you find is, oh, unfortunately, right, this mouse never previously had the opportunity to learn the different antigenic signals of the blue tumor cell, even though it knows the antigenic signals from the red tumor cell. Unfortunately, it does not have protection. Okay. Now, you might say, wait a minute, Dr. Shen, doesn't this imply that we can actually be vaccinated against certain types of cancer, right? What if you injected dead prostate tumor cells into you, right? Would you then become protected against prostate cancer, right? That's kind of chauvinistic, right? Because that only applies to guys. But let's talk about, any type of cancer, right? Liver cancer, lung cancer, brain cancer. We'll include the women also, ovarian cancer, right? Or uterine cancer. Wait a minute, can't you just inject dead cancer cells as a vaccine? No, right? Why? It's not practical because everybody's tumor has its own story of random mutations, right? So even though you have 100 patients, all who are suffering from breast cancer, right, the actual genetic makeup of their cancer cells can be very different from each other, right? So it's just not feasible to be able to say, well, because you're injected with dead cancer cells from this one patient, right? Yeah, okay, great. You're protected now against it in case you're ever exposed to live tumor cells from that patient, right? But it gives you no protection necessarily from other types of cancer developing, in your own, let's say, breast tissues, right? However, that's not black and white. And so when we talk about different types of new immunotherapies, we'll revisit that again, right? And actually see that there are possibilities to vaccinate against cancer, but it's just not as simple as this mouse example here. All right. So the ability of the immune system to detect these cancer types of antigens, right, is a really big part of the ability of the immune system to constantly monitor and nip transforming cells in the bud, right, before they actually get too far in terms of forming tumors. Here's another experiment. Let's say you have two mouse strains here. This dark brown one is a normal, healthy wild type strain. This light brown one is one that's designed or bred to be immunodeficient, right? It has some sort of deficiency in its immune system. In this case, I will just say that these are RAG2 knockout mice. Again, those of you who took immunology are like, RAG, what is that? Ah, the oven mitt, right? Okay, I shouldn't even say oven mitt because those of you who haven't taken immunology be like, what the hell, right? No, right? So these genes code for proteins that are involved in B cells and T cells being able to make things like immunoglobulins and T cell receptor, right? So these immunodeficient mice, they have that gene knocked out, so they cannot actually make things like immunoglobulins and TCR. So these two types of mice are exposed to, again, it's very mean, they're exposed to high levels of some carcinogen, and so they develop tumors, right? Maybe not surprisingly, more of the immunocompromised mice develop tumors compared to the wild type, more healthy mice, right? But it's a strong carcinogen, so these mice develop them as well. Notice these are coded blue and orange, right? What does that mean? These are going to be random tumors. Random mutations are contained in these tumors. Some of these tumors will tend to be more strongly immunogenic, meaning the combination of antigens on their surface are really good, obvious targets for the immune system to recognize, right? Shapes that the immune system loves to see and are very sensitive to seeing. So that means a tumor that is strongly immunogenic is one that is easily seen by the immune system. However, you will also, by chance, have tumors that are weakly immunogenic. In other words, its type of antigens on its surface, right, is harder for the immune system to detect. So the second part of this experiment then is you take some of these weakly immunogenic tumors, right, and then you inject them into another set of mice, the wild type ones and the immunocompromised ones. Right? Not surprisingly, because they were grown in the dark brown mice to start with, right? These tumors, aggressive cancer cells, still can form a new tumor in the dark brown ones, and not surprisingly, because the RAG2 knockout mice are already immunocompromised, right? These tumors also can grow in them. But look at what happens if you take some of these strongly immunogenic tumors, right, and inject them in mice. What you find is that the strongly immunogenic tumors don't grow in the strong immune systems of the dark brown mice. Why? Because the immune systems see them a lot quicker, a lot easier. And so there's a better chance of the immune system in a healthy brown mouse, right, destroying these cancer cells, right? So this would be like what I was joking about, me and my fellow PhD students, if we accidentally injected ourselves with prostate tumors, cells, right? Our healthy immune systems should be able to detect them, right? But however, if the tumors are very or are weakly immunogenic, right, and they have an easier time hiding from the immune system, then you may actually see that some of these blue tumors can grow in the brown mice. Right. All right. So let's look at people now. Right. The effects of a compromised immune system on cancer. So what we're looking at is data here, looking at cohorts of patients who either are HIV/AIDS patients, right? So they already have an immunocompromised immune system due to HIV infection developing into AIDS. And we're also looking at patients who have received organ transplants, right? So when you receive an organ transplant from a donor to a recipient, part of the treatment has to be using drugs to impose an immunocompromised state in the recipient so that the patient's body, their immune system, doesn't outright reject foreign tissue that's coming from the donor, right? So these two groups of individuals we're looking at here, either HIV/AIDS patients or transplant patients who are taking strong immunosuppressive drugs. And then what we're doing is we're looking at the incidence of different types of cancers: breast cancer, prostate, colorectal, ovary, tracheal bronchus, and lung cancers. These are cancers that are not normally associated with any kind of infectious agent, right? These are cancers that largely just kind of develop either through hereditary genetic causes or sporadically through exposure to things like carcinogens, etc. What you find is when you compare the rates of these different cancers in these two groups of patients here, right, that yes, there is some evidence that there'd be elevation of certain types of cancers. So this is a standardized incidence ratio of, how many times you see these types of cancers appearing in these patients in these cohorts compared to the normal population, right? If you had the same number of normal healthy people who are not AIDS patients and who are not taking immunosuppressive drugs, right? So cancers that are not associated with infectious agents, you don't see a huge increase. But what about cancers that are known to be associated with infectious agents, right? Because these two cohorts that we're looking at are immunocompromised individuals, they have a greater likelihood of being infected by infectious agents in the first place. And so not surprisingly, right, the incidence of these types of cancers, like Epstein-Barr virus-related cancers, or Kaposi sarcoma, or stomach cancers caused by different types of fungus in grains that are not properly stored, right? These are more elevated, right? And this really shows you that there's a link between the strength of a person's immune system, right, and the propensity to develop certain types of cancer. So this is just a table that kind of is another look at that table on the previous slide, just looking through different examples of different types of cancers in immunosuppressed transplant patients. All right. Don't have to memorize this. The take-home message is, once again, because different cancers are so variable, right, in their etiology, meaning how they developed, involving different normal cell types, right? Not surprisingly, having an immunocompromised body, right, will have different impacts on whether these different cancers rapidly progress or not, right? So for example, high levels of melanoma patients develop in immunocompromised individuals compared to something like breast cancer, where the rate in immunocompromised individuals isn't necessarily that much more than in expected cases. All right, so here's an example of other immune cells that are involved in fighting the cancer. So we looked before at examples of immune cells that are part of stroma, like the macrophages, that can end up being fooled or tricked or coerced into helping out an aggressive tumor through the activity of macrophages remodeling the stroma. But not all hope is lost. There are other immune cells, right, that can come to the rescue and also actually help fight the tumor cells. So tumor infiltrating lymphocytes, or TILs, recognize bad signals, antigenic signals on the surfaces of tumor cells, and they try to destroy the tumor cells by telling them to undergo apoptosis. So therefore, at the time when a patient has a biopsy, right, if they look under there and actually see high levels of these TILs present in the tumor, that's actually a positive prognostic indicator, because it means the person's immune system is still fighting, right, the tumor cells. So here you can see here's a Kaplan-Meier plot looking at two cohorts of patients. The y-axis is the fraction of patients surviving over the course of many months and years. At the time of first being diagnosed, right, did their tumors show many of these TILs that were there with the tumor fighting it? Or in some patients, were there very few TILs fighting it, right? So very compelling evidence that the immune response is still very relevant to a patient's prognosis. These different antigens that are presented on the surface of tumor cells that the immune system can pick up on. Right? Certain of these antigens are what we call tumor-specific antigens. In other words, different messed up types of signals that a normal healthy cell would not present at all. Right? These might be the result of mutations causing weird misfolded peptides or peptide sequences that are sort of not normal. Right? So those would be tumor-specific antigens. Tumor-associated antigens are actually normal proteins, right, that any cell in the body should be able to express, but perhaps a particular cell type should only express certain levels of it. And if you end up expressing way too much of it or way too little of it, that can also be a sign for your immune system to realize, hey, something's wrong with this cell. Perhaps we should get rid of it. Right? So here's an example. Melanocytes, these are totally normal, healthy cells, express certain tumor-associated antigens, right? Over here. Here are tumor-associated antigens, the MAGE-1 protein, being expressed by developing sperm cells. Okay. Here are some other examples of tumor-associated and tumor-specific antigens, just for you to be able to think about and appreciate. Right? So for example, certain types of breast and cancer carcinomas exhibit levels of HER2 and NU on their surface that are way out of whack compared to normal. Right? This is kind of neat because the third column here shows you particular peptides that come from the protein that's lost its normal regulation, that are the peptides that are particularly sensitive to be used as an antigen for your immune system to pick up on. Right? So these are amino acid sequences from, for example, the HER2 NU receptor tyrosine kinase complex. One way that our cells are very good, one way that our cells are normally very good at communicating with our immune system to let our immune system know that something is wrong versus everything is okay, is of course being able to present things on their MHC platforms on their surface, right? Those major histocompatibility complexes that are in charge of presenting peptides for patrolling immune cells to check out. So not surprisingly then, these MHC platforms are made of proteins, and these proteins are coded for by genes, specifically our HLA genes. So just like any other gene, they can be mutated during the course of random mutations, right? That the transformed cell is kind of stumbling through, sort of experimenting with, right? To see which combination of mutations give it an advantage. So some transformed cells, by chance, shut down the expression of MHC on their surface. And without MHC on their surface, they cannot present peptides that the immune system would use to recognize, right? So this would give these tumor cells a growth advantage because they can avoid being detected. Look here. This is a colorectal tumor in cross-section. Here are the tumor cells at the top, very messy, right? Here are the normal healthy tissues, right? So these epithelial cells are normally arranged. They have a basement membrane around them. These are part of ducts, right? So the white space is the lumen inside of the duct. These epithelial cells are behaving very well. They express a lot of MHC1 on their surface. That's what the brown things are, right? So any immune cells that come by have the opportunity to be able to see what the little MHC1 proteins on these cells are carrying. Right? But look at what's happened to the messed up carcinoma cells over here, right? Not only do they lose their normal shape and organization, that's obvious, right? But look at the level of brown staining, right? They've learned to switch off through mutations the presence of MHC. So these cells, right, patrolling immune cells might not be able to recognize as being wrong. Okay, last slide for the day. These are just a list of different ways that random mutations inside of transforming cancer cells can come up with to be able to avoid or evade the immune system. Right? I would recommend, as you study today's lecture material, take a few minutes and just think about each of these different strategies here, right? These are not things that I'll specifically test you on, right? It's not like I'm going to say in the table, what did it say next to this particular strategy? But I want you to actually understand these and broaden your way of thinking about how mutations inside of transforming cells not only mess up oncogenic pathways, tumor suppressor gene pathways, not only mess up things like fooling the stroma around it to like do a wound healing response, but random mutations can also disrupt these important functions for how normally our immune system expects to see things to be able to identify and eliminate transformed cells. Okay? All right, let's go ahead and stop there today.